Hi, welcome you to this video session. Actually, we have developed a method for pesticide changing in ethyl acetate medium. It is an organic phase biosensing technique. The process involves you extract the pesticides in soft agricultural item using ethyl acetate, then add to it equal volume of aqueous lipase solution of definite proportion, then check for 5 minutes using vortex checker, keep it at room temperature for nearly 2 hours, filter twice with Wattman 1 filter paper, then dilute 3 times and add calculated amount of L-serine to the mixer and then check it for 5 minutes. The solution thus obtained is now ready for bioanalysis using either GST enzyme using Habig's protocol or ACAC enzyme using an Ellman's protocol. If you extract the pesticides in agricultural produce using ethyl acetate and then you add lipase solution to it, then the solution will have two phases like this, upper phase and a lower phase. So initially after mixing we will not see these two phases, but after keeping it for 2 hours this phase separation will take place. Then you filter the supernatant 4 type of materials, get a clear solution which looks like this. Now to this solution you add calculated amount of L-serine and then take it for bioanalysis using either Ellman protocol or by using Habig's protocol. In Ellman protocol, what is done to this mixer, 2 reagents DT and B, that is 5,5-days dithiobis 2-nitrobenzoic acid, and another reagent acetyl thiocholin chloride in presence of third reagent acetylcholine enzyme, esterase enzyme. When you add these reagent mixers in a solution in absence of pesticides, you will get a bright yellow color. It is something like this. It is a white yellow color. It is a solution ethyl acetate lipase and L-serine mixer plus DTNB plus acetyl thiocholin chloride and then acetylcholine esterase enzyme. You will see a bright yellow color immediately after addition of the enzyme. But when pesticide is present in the mixer, depending upon the amount of pesticide present, the color will be faded. The intensity will decrease depending upon the amount of pesticides, and eventually it will lose totally the yellow color when the amount of pesticide is above the expected limit. So in this way, by looking at the intensity of the solutions through a colorimeter, we can know what is the amount of pesticide present in a particular solution. This is based on Ellman protocol, and similarly we can do the same thing with same color type with Habig's protocol. In Habig's protocol, the reagent used are DTNB, that is 2,4 2-chloro to 1-chloro 2,4-dinitrobenzene along with reduced glutathione, and the enzyme GST acts as catalyst to this reaction pesticide when inhibits the enzyme GST, then the intensity of the color, yellow color, decreases in the same way as it was in case of Elmantes. So in that case also, looking at the color difference, we can find out the amount of pesticides present in a solution. Now, determination of pesticides through colorimetric method using Elman's or Habig's protocols is not, of course, new. But novelty of our work lies in the use of solvent and the total procedure. As you have seen, we have used ethyl acetate as the extracting solvent. So far, no report of using ethyl acetate for biosensing using these two enzymes was there in the literature. So we have done for the first time that we have extracted the pesticide in ethyl acetate, and we have made feasible to analyze the same using the two enzyme, either lipase and either acetylcholinesterase or GST, glutathione transferase. So while doing such, the field of applicability of the biosensing of pesticide has got widened. Earlier, this acetylcholinesterase based pesticide sensing was done with water soluble pesticides only, but most of the pesticides used in agriculture are highly soluble in organic medium. So if you want to check the pesticide content in any agricultural produce, use of organic solvent is unavoidable. So but if you use organic solvent, you cannot use the enzyme. So in that way, the use of acetylcholinesterase based biosensing technique was limited to only very selected number of pesticides. But now with the use of ethyl acetate as the solvent, now we can use this testing procedure for almost all kind of organochlorine, organocarbamate and organophosphate pesticides. And similarly, the use of the other technique, GST based technique, naturally the GST based technique is applicable for 6 different classes of pesticides, but we have to do in 25% methanol. But now use of this ethyl acetate based extract has made it possible to test all the 6 types of pesticide in addition to organocarbamate, organophosphate in the same sample. In the same sample, you can use all these mixtures, all the reagents which are responsible for ACAC based test or which are responsible for GST based test. All enzyme you can mix and you can test the presence of all the 6 classes of pesticides in the same solution mixer. So it has made the enzyme biosensing technique more practical, more feasible, and it has widened the scope of its applicability. Moreover, this process is very easy and less cumbersome because now we no need to use the QSR extraction process. The method is almost a robust one because simply by adding lipase, L-serine and by following few filtration steps with ordinary Wattman 1 filter paper, we can apply biosensing technique. Not necessary to follow the rigorous QSR analytical methods. Moreover, this method now can be applied to different classes of pesticides. So in this way, the method is much more advantageous than earlier Elman or Habig's method. Thank you for your attention.